All right, welcome everyone to the March edition of the VanBug Seminar Series. I'm your host, Kara. Before we begin, please everyone make sure that your phones are silent so they don't interrupt the presentation. This is a standard disclaimer that the event is being recorded. So we're going to start with these announcements. We'll move on to our trainee talk, featured talk, and then there'll be some networking at the end where we will provide snacks and drinks. And that will run till 9 p.m. VanBug Organizing Committee consists of 3 faculty members, Dr. William Shao, Dr. Amy Lee, Dr. Faraz Huff. We also have a number of volunteers from both SFU and UBC. We have 2 upcoming events. There is our April lecture, and then we also have a lay term talk competition. This will be in May. This will be open to all undergrads and graduate students. There's a 3-minute hard limit on the presentations, and they need to be accessible and in lay terms. There'll be more details and registration will be available on the VanBug website when we have more details for you. We are coming up to the end of our speaker session. So if there's a particular speaker you'd like to see next year, please suggest them on our website. We take these into consideration when we send out invitations for the following year. We have a number of sponsors for VanBug. We are sponsored by the joint Bioinformatics graduate program between SFU and UBC, as well as Genome BC, Imagia Connexia Health, Langara College, Stem Cell Technologies, and St. Paul's Hospital, where we are right now. VanBug has a number of affiliations with other bioinformatics organizations. We're affiliated with ISCB and the other bug organizations and our sister bugs, MonBug and TorBug in Montreal and Toronto, respectively. Today, I'm pleased to introduce our featured talk by Professor Karin Verspoor. Professor Karin Verspoor is Executive Dean of the School of Computing of Technologies at RMIT University in Melbourne, Australia. She is a fellow of the Australian Institute of Digital Health and a 2021 Brilliant Woman in Digital Health. She was also selected as a finalist in the Women in AI Australia and New Zealand Awards for 2022 for AI in Innovation. Karin is passionate about using data and AI to improve health outcomes for people. Her work has a specific emphasis on the use of natural language processing to transform unstructured data in biomedicine, ranging from scientific literature to clinical texts into actionable information. Without further ado, I introduce our trainee speaker for tonight, Andrew Galbraith. Andrew Galbraith is a master's student in Dr. Stephen Jones' lab at the University of British Columbia. Please come on up.
Oh, William, sorry, how do I connect this to?
Oh, sorry, I forgot to swap it.
Yeah, no worries.
Yeah, I got it.
Great, thank you. All right, hello everyone. My name is Andrew Galbraith, and I'm going to be presenting on my master's thesis, which is genome-wide detection of DNA hydroxymethylation in various cancer types using nanopore sequencing. Okay, so just get a brief overview of my thesis project. Well, essentially, my aim is to detect hydroxymethylation in around 180 personalized oncogenomic patients enrolled here at BC Cancer using nanopore sequencing. And essentially what I'm trying to do is find unique 5-HMC profiles in cancer. So one hypothesis we have is that epigenetically mutated cancers will have a different hydroxymethylation profile than non-mutated or non-epigenetically mutated cancers. We also aim to see 5-HMC profiles across cancer types, biopsy sites, primary to metastatic tumors. And we want to particularly look at these hyper and hypohydroxymethylated tumors to see if we can look backward to see some kind of genomic or transcriptomic signature in these tumors. And this is where the multi-omic framework of kind of our personalized oncogenomics approach here comes into play, where we have genome and transcriptome, as well as methylation data that we can compare to for any findings I make. So what is hydroxymethylation? Well, briefly, it's an intermediate in the process to demethylation. So you can imagine here, we have our cytosine in a promoter of a gene, and when we methylate that cytosine with DNMT, we'll get a methylcytosine, and methylation in promoter regions is associated with silencing of the gene. And methylation is a stable epigenetic mark in around 60 to 80% of CpGs. But when a TET enzyme comes in, it can actually add a hydroxy group to the methylation, and this leads to a process of active demethylation. Interestingly, though, hydroxymethylation is not just an intermediate, but a rather stable mark that occurs at around 2 to 10% of CpGs at a rate 10 to 100 times higher than any of the other intermediates in the pathway. Also, TET is not the only enzyme implicated in this pathway, but also the citric acid enzyme, IDH1, plays an important role, where it creates alpha ketoglutarate, which acts as a substrate for this pathway. And if you have a mutated IDH enzyme, you're going to have a failure of this pathway to occur. It's going to get inhibited. So next, how has hydroxymethylation been implicated in cancer? And generally what researchers have found is a global loss of hydroxymethylation in cancer. And this is due to regulatory mutations in the cancer, as well as oxidative stress in hypoxic environment the cancer is exposed to. But this can be region specific generally. So again, some areas of the cancer might be hyperhydroxymethylated, but overall researchers have found that the malignancy of the cancer is inversely proportional at some regions to your hydroxymethylation. And this finding can be both diagnostically and prognostically predictive. So researchers have used hydroxymethylation signals to diagnose cervical cancer, colorectal cancer, gastric, pancreatic, liver, and thyroid cancer. And this hasn't been applied to a clinical framework yet, but just on a research kind of analytics basis. And they've done this using both genomic and cell-free DNA. Now getting into nanopore sequencing. So essentially, we use nanopore sequencing to detect hydroxymethylation in the same way it detects a base. So again, you get these peaks in your current through your nanopore, which you can use a neural network to infer whether that's a cytosine or a thymine. But you can also infer whether that's a methylated cytosine or hydroxymethylated cytosine. So we use this neural network model, Guppy, which does this for us. And then from it, we can get the frequency of hydroxymethylation at given CpGs. And listed below are just some other methods of finding modified bases, such as oxidative bisulfite sequencing, immunoprecipitation, or PacBio sequencing. So what are the advantages and disadvantages of our nanopore approach? Well, the main advantage here is the long reads, and these really enable better mapping across the whole genome, so we can do global detection, as well as one interesting thing they do is we can do haplotyping with these long reads. So for each pod case, we actually have allelic hydroxymethylation and allelic modification calls, and we can look at differential allelic hydroxymethylation in the same cancer sample. We also, with nanopore, can call the DNA sequence and modifications in parallel and ultimately get whole genome base pair resolution of hydroxymethylation. The disadvantages of nanopore is it's still not the most accurate sequencing technology, and largely, especially for identifying hydroxymethylation, this is still in development, and there hasn't been any sort of review papers validating the models we've been using for our pipeline. So here, this is the validation data we sort of have, and this is from a nanopore talk, so take it with a grain of salt that they're probably just presenting the best data they have. But when they did test nanopore in the model we use in our lab on synthetic hydroxymethylation, They found that the chemistry we use in our lab was 93% accurate in calling hydroxymethylation. However, when we did a comparison on one sample of hydroxymethylation to oxidative bisulfite sequencing, we actually only found a 10% correlation. However, there were limitations with this analysis, such as sequencing depth, the reliability of our oxidative bisulfite calls, which we were using as a standard, as well as the sparsity and low frequency of 5-HMC calls, which makes it really difficult to get a high correlation. So how does our pipeline actually work to call hydroxymethylation? So essentially, we have our nanopore FAST5 files. We're going to run Guppy, which will simultaneously call the bases and the modifications while aligning it to a reference genome. Then we're going to merge, sort, and index our BAMs. We're going to use WhatsApp to haplotag our BAMs so we can do allelic calls. And then we're going to extract our hydroxymethylation and methylation calls. And so I started my master's in the fall. And so I haven't finished running this pipeline on all pod cases yet. But so far, I have done a preliminary analysis of 36 pod cases, 18 of which were epigenetically mutated, 7 having TET mutations, and 5 having IDH mutations across 18 total cancer types. And so one thing just to note, looking at these figures, there's a lot going on, but I just wanted to pick out how much variation we actually see across cancer samples with hydroxymethylation. So take, for example, all these light green bars. That's the same cancer type, each with no epigenetic mutation, and yet we still see so much variation in those hydroxymethylation calls. If you look at the figure 1 over, now this is mean hydroxymethylation frequency across all CpGs globally. Even in cancers that have the same epigenetic mutations, there is a lot of variation in hydroxymethylation. And overall, if we're comparing cancers with no epigenetic mutation to cancers with epigenetic mutation, we don't really see a lower distribution in those epigenetic mutation categories, mostly due to how much variation there is across the whole entire distribution as a whole. However, we can see in the tumors with no epigenetic mutation, there does tend to be more outliers that are hydroxymethylated, which we'll talk about later. And then comparison to methylation data, you do see a little bit more of a clear distribution in the methylation data with the epigenetically mutated tumors. And this is across different categories, such as if it's a chromatin enzyme which is mutated, a DNA enzyme that's mutated, or a DNA and a chromatin enzyme that's mutated. But you do see a little bit of a higher distribution in those cancers of methylation than in the non-enzyme or epigenetic mutated cancers. And now getting into some box plots, I just wanted to show the overlap in the distributions here. So our hypothesis coming in was that in these epigenetic mutated tumors, especially in the DNA epigenetic mutated tumors and the TET mutated tumors, that we would see a lower distribution in hydroxymethylation due to inhibition of the demethylation pathway. However, we didn't really see this in the small cohort that I've analyzed so far. So as you can see, there's a large overlap across no epigenetic mutation to different epigenetic mutation categories. And even in TET mutated cancer samples, we still see overlap in hydroxymethylation with tumor samples with no epigenetic mutation. If you're looking at the methylation data, you see a little bit of a higher distribution of methylation in tumors which have an epigenetic mutation. I also took the average hydroxymethylation and methylation at CpG islands and wanted to see if you could cluster tumors by cancer type based off the hydroxymethylation profiles at these islands. However, you can see the clusters are largely overlapping. And also for hydroxymethylation, we have a line distribution, which is due to the sporadic low frequency calls of hydroxymethylation. So if I was going to redo this analysis, I'd want to transform the hydroxymethylation data first. In the methylation data, you can see some clustering by cancer type. However, the clusters are also a lot of them are overlapping. One thing I wanted to look at too is for some cancers, we actually had multiple samples. So I wanted to assess the variation across samples from the same cancer in the same individual. So again, here are 3 individuals, each with 2 sample points taken for their tumor. And you can see there's quite a bit of variation in the samples from the same individuals. So POG-B liver and POG-B liver taken at 2 different time points have more variation between each other than between an individual with a different cancer type, looking at POG-B liver to POG-C liver. You can also note in this that in the methylation plot, you can have really big within intra-individual variation due to biopsy site, which I think gets at how epigenetic differences in tumors can be very region specific. So you can see in the same patient, the same cancer, if you take that cancer in the spine, its epigenetic profile is going to be completely different than if you take the same cancer from a lymph node. And it could be even more different than how much variation there is across individuals with different cancer types. Since we didn't get the results we were quite looking for, one thing we considered is we were looking at the nanopore talk data, and you can see there's a little bit of a bleed through from methylation to hydroxymethylation, where the greater the methylation frequency, you're going to see a greater hydroxymethylation call. And because there's such a class imbalance when you were looking at whole genome, because the rate of methylation is 10 times higher than the rate of hydroxymethylation, we thought one simple way to maybe account for this would be to look at the rate of hydroxymethylation over the rate of methylation. And overall, we see it gives a little bit of a better signal. So now looking at globally and at CPGs, we can see the distribution in these epigenetically mutated tumors is slightly lower than in the tumors with no epigenetic mutation. And also in the tumors with no epigenetic mutation, there are some clear upper bound outliers, which are hyperhydroxymethylated. And you can also see in the TET mutated cancers as well, they tend to have a lower distribution in hydroxymethylation. So findings and next steps. So overall, we didn't see a clear notable reduction in hydroxymethylation in these epigenetic mutated tumors, but there's a lot of confounding factors at play here, which are going to add extra layers of variation, such as our sample size. We had multiple cancer types in this original cohort. Some cancer types only had 1 sample for 1 whole cancer type, right? So we really need to get more samples for each cancer type so we can properly control for that factor. Same with looking at the factor of biopsy site, and even factors such as the tumor microenvironment and the technical conditions are going to add extra layers of variation, which we can better account for with a larger sample size. Also, with these epigenetic mutations, I took both missense and nonsense and indel mutations, so that the mutation effect here isn't always certain. You can imagine some missense mutations in the context of a cancer cell are probably meaningless. And so even though we're saying there's a TET mutation here, we actually don't necessarily know if it's going to have a phenotypic impact. Again, I've talked about this a little bit, but the methylation calls are bleeding through into the 5-HMC calls. So we may want to think of a better way to account for that in calling 5-HMC. So the next step here is really to do this analysis on a much more comprehensive set of these cancer patients, do region-specific analysis. So we're not just looking globally, but we're doing differential hydroxymethylation analysis to find specific regions which are enriched in certain cancer types. Next, instead of just looking at the epigenetic mutation, let's look at epigenetic dysregulation through RNA-seq, because we have that data. So instead of looking at if it's TET mutated, let's look at the levels of TET expression and see if there's different hydroxymethylation. And then finally, really looking at these hyper to hypohydroxymethylated tumors and trying to find a unique signature for them. And this is a sneak peek to the future results. So Right now, I'm up to 90 pod cases that the pipeline has been run on. This is for the first 79 here. And here we have mean hydroxymethylation over mean methylation. And I wanted to note that, again, lots of variation across cancer type. You see these hyperhydroxymethylated tumors. Some of them stand out quite a bit as outliers to the rest of the distribution. And also, if you look at the presence of epigenetic mutations in these tumors, again, you generally see in these hyperhydroxymethylated tumors, no epigenetic mutation, except for one here, which has a chromatin mutation, which we wouldn't really expect to be relevant for hydroxymethylation anyways. Whereas if we look at the 10 most hypohydroxymethylated tumors, 5 of them have a TET mutation. These are my references. And if you have any questions, now would be the time to ask.
Yeah, so you're trying to cluster now on based on like cancer type, right?
Yeah.
And also like biopsy sites there. What's the variation like within a tumor? Is it so like in bulk tissue, right? Clustered by cell type, but is it so like homogeneous or just so randomly varying that you wouldn't expect that to be a major component of variation?
One thing, again, there's no possible way to, like, at least in our framework right now, to do like single cell hydroxymethylation. And so I think a lot of that's up in the air and unknown a little bit. But one thing we are looking to account for is just tumor content in these bulk sequencing samples too. So that's another factor that we'll be looking at too. Because again, yeah, you're gonna have intratumoral heterogeneity. Right now, I don't think we can really account for that. But what we can look at is how much of the normal cells might be in, or the normal cell DNA might be in a specific sample, and then see if that's playing a confounding role at all.
That leads nicely into my question. I was going to ask, I know you said that there's a methylation seems to be different between different biopsy sites. And I was wondering if the difference in methylation, how much you think that's influenced by having a lower tumor content reliably at some biopsy sites? Like, would it be influenced by the normal cells?
Yeah, I think it definitely would. So that would be another interesting thing to look at as well. But I do think, like, even if they had the exact same tumor content, again, especially epigenetic differences, a lot of it is, from what I've seen in the literature, like environment specific. So the tumor's epigenetic profile is really going to adapt to the specific environment it's in. And then, yeah, there's going to be quite a bit of variation because of that.
All right, if there are no other questions, I think we're ready for our featured speaker.
Sorry.
Nice.
See if we can wake this thing up. Aha. There we go. Okay. Hi, everybody. So as you heard, I'm Karin Verspoor, and I have come all the way from Melbourne, Australia. And I'm really happy to be here. Thank you very much for the invitation to be here to speak with you. It's been a pleasure to be visiting here and to learn all sorts of things about what's happening around Vancouver. So yeah, thank you again. So today I'm going to talk to you and give you a bit of a sort of high-level overview of a body of work that I've been doing for a couple of decades now, which we were discussing earlier. It sounds amazing that I've been doing this for so long, but it's been quite a journey. And to be honest, there's still so much to be done and still so much to be learned. So hopefully I can inspire you all a little bit to do some of this work yourself, because I can't do everything. So I'm going to be talking to you about the use of AI and natural language processing in particular for looking for evidence in biomedicine, and also synthesizing that evidence and ultimately supporting discovery of new evidence. Why do we care about this? Because, at the end of the day, all of our clinical research activity really aims to understand things, right? To contribute to our knowledge and understanding of particular interventions and their efficacy, particular molecular reasons for disease. We want to be able to understand how things work, why they work, what works, I guess, at a fundamental level. And that drives the entire activity of science, basically, right? So we learn things, and then we ask questions, and then we learn new things. And so all of this is ultimately about improving the way that we can treat people and understand, tumors and detect prognosis and that sort of thing. If we take a step back and sort of ask, okay, what is evidence? Why are we trying, we're trying to collect these things. What is evidence, though? Ultimately, evidence is really just scientific conclusions, right? So, we heard in the previous talk that we're comparing different scenarios. We're doing analysis. Ultimately, we're trying to come up with statistically meaningful conclusions across a population. And we do that by studying things and asking questions about that, and doing controlled experimentation where we can really explore the impact of different factors at a fine-grained level. And what we have in the clinical context, in the medical context, is this very sort of well-developed notion of evidence-based medicine. So we rely on this evidence base and these scientific conclusions that are driven by the entire clinical research activity to inform the way we treat people, right? So every decision about what drug to treat a particular disease with is grounded, we hope, in some scientific evidence and some conclusion, some study that's been done. And at the moment, our gold standard of evidence really is randomized controlled trials. And we prefer when we've done this across multiple trials, and we have the ability to show that these results generalize across different trials and different populations. But ultimately, we're relying on these scientific studies in order to inform our next steps, right? And inform the way we do things. And this is done typically through very controlled selection of cohorts, right? So we have narrowly defined inclusion criteria and very strict exclusion criteria. And fundamentally, what we're talking about here is the scientific method, and building on that in order to drive our understanding of how things work. All of this information that we're capturing, all this evidence that we're capturing, could be going into databases, and it could be going into sort of shared repositories of information. But the primary place where we find this is actually in the research literature. So in scientific publications that describe these controlled experiments that we do, and then are published and made available for other scientists to learn from, and ultimately then to translate that into actual sort of clinical use and decision making. And so we can view the research literature, the scientific literature, as the primary repository of everything we know in science. And if you ask yourself, where else does this information live? I think you probably reflect that it's nowhere else, right? Everything we know about science has to be communicated through publications, or it's not actually known, in the sense that it's not known across a broad community. So what this means is that the research literature is really the best place we can go for evidence, because it's sort of, it captures everything we know about science. And what we want to do is think about how we can leverage this literature to do new science, right? So if you think about the talk we just heard or any of your own research projects, where do you start? You start by doing a literature review. That's so that you can understand what we already know. And then use that to identify a gap, right? A question that you want to then answer in the next step. So we can think of the literature as the key source of knowledge that drives science and the way we progress and understand things. So what I want to talk to you about is how we can basically help that process along ultimately. There's so much information that's captured in the research literature that it becomes paramount for us to develop tools and methods for extracting that knowledge and helping people do this process of science. And that's really, in a nutshell, a very tiny nutshell, what I've been trying to do for the last couple of decades. So let's talk a little bit about detecting evidence in the scientific literature. So as I told you, one of the key frameworks that we have for medical evidence in particular is randomized controlled trials. And the basic structure of a randomized controlled trial can be summarized in terms of a model called PICO. The patient of interest or the population of interest, the intervention that was studied, the comparison or control group that is used in a randomized controlled trial, and the outcome that is investigated. And so you can summarize almost any randomized controlled trial through a statement like, we are aiming to assess the effects of some intervention compared to some comparison or control group for a particular condition or problem in a particular population in some context on outcomes. And almost every clinical trial that follows the randomized controlled trial structure has this sort of statement. And of course, then the problem becomes, well, how do you characterize the intervention? How do you characterize the population? But in a nutshell, everything boils down to this. And so one thing that we can do is to look through the literature and basically try to identify those key elements. What's the population that's described in a paper? What's the intervention that's described in a paper? What's the outcome that's of interest in the paper? And so there's been a body of work around trying to find mentions of populations, interventions, and outcomes in research literature in order to organize and structure that information. Because it's a lot easier to deal with a little template, oops, sorry, wrong way, a little template that summarizes the population, the intervention, and the outcome associated with the scientific contribution than it is to read through all the words in every paper out there, right? So if we can identify these key elements, then we can organize that information and make it easier to find and easier to draw conclusions from at a higher level. So there's been work to try to detect these elements. And here what I'm showing you is actually a picture of a corpus that was developed by Byron Wallace and his group called the Evidence-Based Medicine NLP corpus. And they've gone through and annotated the PICO elements or the PIO elements. We tend to kind of ignore the control group for this purpose across a number of articles, which we can then use to train natural language processing models that can detect these in many more articles. And so the objective of what we're trying to do is basically build a little tool that effectively machine reads a whole bunch of publications. Remember, in PubMed, we have something like close to 40 million articles in the scientific literature that have been indexed there now. So like, I don't know how many papers you can read every day, but I probably can't read more than one. So, even over the course of many, many decades, you're not going to come anywhere near reading everything that's known, right? So, but computers don't get tired and they can process stuff at scale. So let's build one of these models and let the models read the papers help us organize the information. Now, that's not to say that you're not going to go and then read these papers if they're relevant to your particular area of interest. But you want to, and I recommend you don't rely entirely on these tools. They're not perfect, but at least it can point you in the right direction and make it easier to find the things that are relevant to your question. Okay, so we can detect the PICO elements. We can also detect more fine-grained kinds of information. And so there's a vocabulary resource that's commonly used in biomedical context called the Unified Medical Language System, or UMLS. And there are various tools out there, including this one from the National Library of Medicine called MetaMap, which allows you to identify particular clinical contexts that are described in the text. So going beyond the population, the intervention, and the outcome elements of a clinical trial, going into phenotypes and signs and symptoms and drug names and much more kind of detailed level of the concepts that are in there. And so now we have tools that allow us to capture population descriptors, right? The P in PICO, but also more fine-grained aspects of disease names and interventions corresponding to drug names and all sorts of things in there. And so again, we can run these tools over vast quantities of literature and really get a fine-grained representation of what's in those papers. So during 2020, I don't know what most of you were up to, but I sat in my room thinking, at home, of course, like everybody else in lockdown in Melbourne, thinking how I could leverage the tools of natural language processing to help gain insight into what was happening with COVID. And we started with this notion of detecting concepts and wanted to try to build a representation of the scientific literature that would allow us to start kind of doing deeper level characterizations of the information that was out there. And so we built a system that combined this PICO detection with the UMLS concept detection. Nothing novel, okay? We're just taking existing tools and we put them together so that we could then represent the literature in terms of these things that I call PICO concepts, which are basically finer grained characterization of the outcomes, the interventions, and the populations in terms of these more specific concepts that we were able to identify using the UMLS. And actually, we specifically used the medical subject headings subportion of the UMLS just for simplicity purposes. So what we did was take a large document collection, in this case, a COVID document collection, and ran it through these different tools in order to identify both the PICO spans and the more specific medical concepts within those spans. And we end up with, taking a sentence like this one: cumulative COVID-19 related hospitalization and death rates. We've recognized that whole phrase as an outcome. And then we've said, okay, what are the more specific outcomes that are captured within that phrase? Well, we've got hospitalization as one outcome and death rates as another outcome. And so we can capture at that finer grained level, more specifically, what's the outcomes that have been studied in this particular paper. We can run that at scale and then build a database which captures and indexes papers according to those PICO concepts. And I'll come back to that in a few minutes and tell you a little bit more about what we did with that. I wanted to wind back to some work that I did a long time ago. So this was work published in 2012, where we were similarly trying to use the tools of text mining and natural language processing to go into some molecular level detection of things. And in this case, we were trying to essentially identify specific functional sites in proteins. And you think, okay, why would I want to go into the literature to find evidence of functional sites? Well, it turns out that there's lots and lots of discussion of molecular biology and particular functional sites in the scientific literature. And in this case, we actually combined a molecular modeling approach called dynamics perturbation analysis, which was developed by one of my colleagues when I was at Los Alamos National Lab, with text mining. And I call this, by the way, brain dead text mining, because all we did was actually look for mentions of residues in papers. And the basic hypothesis there was that if there was a residue that was mentioned in a paper, it was because that residue is important, right? Very simple idea. Why would you bother mentioning and calling out a specific residue in a specific protein if there wasn't something going on at that protein? At that residue. So what we did was we found all of the mentions of specific residues in texts, at scale, across any reference that was mentioned in the protein data bank, into PubMed. And we recognized specific residue mentions using very simple, like I said, brain dead text processing patterns. And we then basically said, okay, it was mentioned, therefore it's interesting. And then we finally overlaid that with the predictions of this dynamic perturbations model, and essentially said, if we have a coincident residue that was identified both through the predictive modeling and the text mining, then this is an interesting place to look. And I'll save you all the details of exactly what the results of this are, but just to give you a sense of what I mean by saying we're going to do text mining to detect the particular residues, we're looking very specifically for things like Gly17 or a mutation like substitute serine for proline at position 97, ARG66, Glu70, cysteines at position 642, 48, like these more complex, they're obviously, we're going to identify the basic amino acids and the positions, but we also want to deal with some of the more linguistically interesting ways of expressing this kind of information. So, we just wrote some very simple patterns that would capture these different ways of expressing the residue mentions and apply it at scale across the PDB, and any references that were captured in the PDB. We then essentially try to show enrichment effectively of particular residues and identify those as interesting from a functional site perspective. And so here's just one example with the yellow bits of this picture correspond to the predictions of the dynamic perturbation analysis model, as this is where the sort of functional sites are within the structurally within the protein. And we find that these sort of the magenta and the red bits are the mentions of the residues that were identified in the papers. And we found a lot of coincidences. So again, anything that was mentioned, it was very, very common for residues that were mentioned specifically in the literature to coincide with the predictions of dynamic perturbation analysis. And ultimately, the work showed that this assumption of any mention is meaningful, basically, was really helpful for kind of pinpointing the particular residues that were functionally active. So we can take this kind of basic paradigm of looking for patterns and looking for concepts and looking for entities further and start to ask all sorts of questions about what is in the literature. We can capture protein-protein interactions. We can look for chemicals that treat a disease or mutations that cause a disease. And we can transform basic sentences that describe these things into a more structured representation, right? So if in the literature, you have a sentence like cyclin E2 interacts with CDK2 in a functional kinase complex, we can detect, okay, there's a protein-protein interaction described in that sentence. It has two interactors. They are specifically cyclin E2 and CDK2. And moreover, we can label that entire interaction as an example of a polyphosphate kinase complex. And so we can map it to a gene ontology term that summarizes the sort of functional information associated with that interaction. We can also incorporate some knowledge into this process so that if we're looking for a chemical treating a disease and we see a sentence like the ACE inhibitor treats hypertension. Well, hypertension we can clearly recognize as a disease. ACE inhibitor is not a chemical. It's not a drug. It's a class of drugs. And if we know that benazepril is an ACE inhibitor because we have a knowledge resource that classifies our drugs, then we can detect that ACE inhibitor treats hypertension is in fact an example of chemical treats disease, even though it's referring to the class of chemicals rather than the specific one. So we can build on this paradigm of detecting concepts to have a little bit of abstraction or generalization embedded in. But the basic idea, no matter what kinds of relationships and entities we're looking for, is about trying to find specific entities, specific concepts, and connect them together in particular ways so that we can structure the information that's there. To give an example, this is from the Kraft corpus. We were talking about this earlier. So this is an example from the Colorado Richly Annotated Full Text Corpus, which when I was at the University of Colorado School of Medicine, we developed a heavily annotated full text corpus of particular biological concepts from ontologies, including the cell ontology, the protein ontology, sequence ontology, and the gene ontology, KEBI as well. And we did heavy annotation of about 97 articles. And then we developed tools that will allow us to recognize all of these ontology concepts and use the Kraft corpus to evaluate our ability to do that. Now, none of this is using machine learning. So again, fairly simple sort of dictionary lookup techniques, but the power is in what we produce and the grounding of particular words in ontology concepts. Along the way, we do need to have a bit of sophistication. We can't just look up dictionary terms because we also need to be able to account for some linguistic variation. So, of course, if we have terms with associated synonyms, we can capture those in a kind of direct match, but we also want to allow for some linguistic variation so that membrane budding also can be recognized as occurring in a sentence that talks about budding of vesicles or vesiculation process, rather than the explicit words and terms that are in the terminology resource. So we do have to have a little bit more than just dictionary lookup, but how much more will vary depending on the complexity of the term space that we're dealing with. Here's another example. So we can take a title and abstract like this one, which refers to, sorry, I'm really bad at pronouncing these chemical names, but propylthiouracil, maybe something like that, induced hepatic damage or induced liver damage. We can apply these concept recognition tools to identify the chemical name, the disease's names, and then transform this text into a structured representation of a particular relationship. So in this case, we're looking at the relationship between something something that we've normalized to an ontology identifier. In this case, it's the ontology identifier for the chemical propylthiouracil, and the ontology identifier for both liver damage and chronic active hepatitis. And so we can boil down all of these words here to a structured representation that captures that basic relationship between the chemical and the diseases that are mentioned in this particular title and abstract. So hopefully you get a sense of the idea. To give you a little bit of a sense of how we approach this, back in the days before there were large language models, what we did was actually do a lot of analysis of the syntactic structure of the sentences. So we're actually building little grammar machines that try to understand what's the subject of the sentence, what's the main verb of the sentence, what are the prepositional phrases, and how are they connected to each other. And then we built machine learning algorithms, which would essentially look for similarity between training examples that we've seen in a corpus that's been annotated with these entities and relations, and try to generalize the patterns, the linguistic patterns that are captured in those sentences, so that when we see another example of a particular relationship, like a chemical induced disease relationship, or in this case, the cause of a seizure, generally the cause of the phenotype, then we can find new examples. So we're always trying to build models that learn from annotated data to then apply those models to data we haven't seen before, so that we can find the basic patterns that are implicit there. And yeah, this was work done in 2013. So again, work long before we had very sophisticated machine learning models to help us do this sort of thing. And we don't have a lot of annotated training data, so this also limits what we can do with sort of very sophisticated machine learning. In more recent work, we're also doing this in the chemical patent space. And so we're now going from biology to chemistry, but again, following exactly the same process. So we're trying to extract the evidence from these patents that describes the entities and the relationships between them, and specifically in this case, trying to identify the roles of entities in particular chemical reactions. So we're trying to identify reactants and solvents in products. And we've been running a challenge, actually, to the natural language processing community to develop methods and solutions for this particular problem and associated problems, called CHEMU, which stands for Chem Informatics Elsevier Melbourne Universities, but also emu is, of course, a very cute little Australian bird animal. So we have our chemical emu over there. This was a challenge test that we ran over the course of 3 years, in fact, and we had a number of teams from around the world actually using our data, which we spent significant resources along with Elsevier to annotate, and then asked people to build named entity recognition and relation extraction systems that would be able to reproduce the annotations that we did. We also handled a number of other cases, which I can tell you about another time. But this is again, the basic idea is that we're running through these texts, trying to find the chemical entities in this case and the relationships between them in order to transform what is a pretty complex text into a much more kind of easier to manage set of information, where particular text spans are associated with particular entity types, and we have relations that are captured between them. So everything I've told you about follows the same paradigm of identifying entities, identifying relationships between them in a targeted way for a particular kind of evidence extraction, but we're really trying to transform the large body of text information, which is hard for us to deal with, into structured data that we can then capture in databases and use for all sorts of purposes. One of those purposes will be search. And as I mentioned to you, I started playing around with COVID-19 data in early part of 2020. And one thing that I realized at that time was that we had a very different kind of search need than we'd had until that point. So if you think about when you go to PubMed and you search for an article, typically you look for something very specific, right? You look for this particular mutation in the context of this particular disease, and you're looking for very kind of specific things. If you wind your head back to 2020, we knew nothing about COVID, right? Literally nothing. And all we knew is that there was this thing, this virus called COVID, SARS-CoV-2, which had been identified as causing disease. We didn't even know what the symptoms were. We didn't know how to identify somebody with COVID. We absolutely didn't know how to treat it. And so there were actually a number of questions that were posed by the World Health Organization at that time, which were extremely open-ended. Okay, what has been published about medical care for COVID? What do we know about vaccines and therapeutics for COVID? What do we know about the risk factors for COVID? If you go into a search engine or you go into PubMed and you type in COVID-19 medical care, what are you going to get back? Nothing, right? I mean, well, everything and nothing, probably nothing meaningful, because it's not specific enough in the context of PubMed for you to be able to meaningfully navigate the results. So it turned out that there was a huge amount of research that was happening in the very early days of COVID. So if this is a snapshot of Lit COVID, which was created by the National Library of Medicine in the US to capture all of the research that was coming out about COVID in PubMed. And if you look here, like as early as I'm not really sure what's going on here, but anyway, as early as January of 2020, we already started to see research coming out in PubMed that was talking about COVID-19 or SARS-CoV-2, and then it really ramped up quite quickly and then remained relatively stable. But just look at the y-axis here. In May of 2020, we already had 2,000 articles per week being published about COVID-19. Now, again, how many papers can you read every day? And if your questions are really open-ended, how are you going to find anything? So I started thinking about how we could solve this search problem. And I actually had a great conversation with a journalist at Science who discovered text mining, as I like to say. As you know, I've been, and it says there in my quote, I've been building these kinds of tools for 20 years, and now all of a sudden there's an interest because guess what? COVID happened, and people can't find papers that are relevant to their questions. Okay, so if we think about the typical kind of search model, we have these targeted queries, we have a well-defined information need, we know what we want to find, right? We know what we're looking for. And we put that into a search engine, and we get back some snippets, and we get back some pointers to papers that mention our terms, and we have very good ranking. Google search. I know my sister's a neurologist, and she tells me she never goes to PubMed. She goes straight to Google. These algorithms work really well when you know what you're looking for. There's, of course, a limited depth in the search because you generally don't go past the first page. So who knows what's out there that you haven't looked at? But anyway, that's what you're going to get. And you're going through one by one and looking at this. Now, in the context of this information need in COVID, I think this kind of process of formulating a very specific query with very particular concepts becomes problematic because we just aren't looking for something that's specific enough. And so this kind of approach, which this is a very typical sort of thing that you see in a systematic review, where they define the very particular mesh terms or title abstract terms that they're looking for in order to identify relevant papers. So this is a very kind of typical search process for a systematic review. But I didn't think that was good enough, and it wasn't going to help us with this COVID-19 problem. And so I started thinking about these categories and these concepts and these terms and these relationships that I knew we could pull out of these papers if we only tried. And so I started thinking about how we could use natural language processing to organize the information that was out there and help people find things. So we developed a system that I call COVID-C, the Scientific Evidence Explorer for COVID, which actually pre-processes the literature in terms of those PICO concepts I told you about earlier, and then provides a visual summary of the literature in order for people to find things that they may not have anticipated. So we took those PICO concepts, we identified population concepts and intervention concepts and the more specific terms that sat under them. And we organized them into a visualization of what was out there. Now, this was done quite early. The screenshot was taken quite early. But the idea is that, you could put in a fairly generic search into the search engine. And then you would get back a visualization like this, which would tell you what are the population concepts that were identified in that subset of literature. What are the intervention concepts that they were then connected to, and what are the outcomes that people have been exploring? Now, there's a bunch of mess in here because the PICO concepts are actually kind of messy and not that great. But this was really just a proof of concept to support a different way of navigating the literature, which was facilitated by recognizing concepts and recognizing PICO elements in this very complex and large body of literature. So it gave a way for people to explore the literature that was rather novel. Okay, so the other thing that we might need to do with evidence is to synthesize it, right? So one of the problems that we've been working on is actually supporting the use of evidence from the literature in the context of clinical decision making. And the way evidence-based medicine tends to work is that we develop guidelines based on the research literature, which advise clinicians about the best practices for particular clinical scenarios. And a key part of this process is actually doing quality appraisal. So understanding how reliable effectively those scientific conclusions are. And as I told you, the clinical questions are typically in this PICO format, and we will do a systematic review by collecting all of the studies that are relevant to that PICO question, and then summarizing that, doing meta-analysis, and typically doing a quality appraisal process along the way. So we can, when we have a particular clinical question that we're trying to understand, so we want to know, what is the impact of hormone replacement therapy in postmenopausal women on cardiovascular risk. We want to understand it decreases or increases that risk, and so we need to make a decision about whether or not to prescribe hormone replacement therapy in this particular context on the basis of the evidence that's available in the literature. And we want to make a recommendation based on the quality. So this needs to be done through an analysis of a large body of literature, all of the literature, hopefully, related to a particular kind of PICO question or a clinical question. And we need to have some way to measure or estimate the confidence that we have in the findings that we have there. And there's a framework called GRADE, which aims to characterize the quality of evidence in the body of evidence, the body of research around a particular clinical question. And so we set about trying to estimate the quality of evidence using the information that was captured in a systematic review in order to allow us to predict the quality and essentially how much we should trust that body of evidence around a particular clinical question. So we've been doing some work around rating the quality of evidence for particular outcomes that are captured in systematic reviews, so that we can inform the development of best practices in the field. So in this framework, where you start with the sort of basic assumption that following the scientific method using a randomized controlled trial gives you good quality evidence, right? That's the whole foundation of our paradigm here. But we then look for information that implies that we should downgrade or trust less the information that's available to us from those studies if there is a risk of bias in the studies, or if there's an inconsistency between two studies, or if there's some imprecision in a particular result that's been demonstrated in a publication. And so the grade framework is a framework for humans to read through these systematic reviews and come up with this meta-analysis that kind of draws a conclusion about the quality of evidence around the particular clinical trial, clinical question of interest. And this informs the recommendation process. So we did some analysis of the sort of the quality of evidence around clinical questions, and found that the medical evidence that we actually have and that ultimately inform our decision making is of high quality only about 1/6 of the time, and half of the time the evidence is of very, very low quality. And so we need to understand basically what's good evidence and what's bad evidence, and we need to have tools that can help us identify this from the literature. Yeah.
We usually take the question to the end, but I figure this one. So just want to clarify, is each of the unit that you're measuring a systematic review or the evidence?
It's the papers described within the systematic review. Yeah, and systematic reviews are typically organized around particular clinical questions and some variants. So it's within the clinical question.
So the review itself might be high quality, but it might have cited a bunch of low quality evidence.
Exactly. That's exactly right. Okay, so a bunch of machine learning, computer science stuff, maybe don't worry about it too much, but basically what we tried to do was to take the information that's available from within the systematic review and to come up with a way to automatically score the quality aspects of the body of evidence that was captured in the systematic review. Oh, that's okay. Pizza, very important. Actually, I have a funny story about pizza and talks, which is that one day I was sitting in my office at the University of Melbourne, and the student came by and he was looking very lost. And because I'm very helpful, I said, "Hi, can I help you find something?" And he was like, "I'm looking for the pizza talk." I was like, "Do you mean the computer science seminar?" And he just looked at me blankly and said, "The pizza talk." And I was like, "Okay, I think there's pizza at the computer science seminar. So I think that's over there." And anyway, it was just funny. Forever in my head, pizza talk is like, it's a thing, right? And I figured he was a starving student and he only cared about the fact that it was pizza. He had no clue that it was a computer science talk. He didn't care. He just heard there was going to be free pizza. So anyway, pizza talk. Okay, so anyway, back to the pizza talk. We were trying to build an automated method for scoring these grade elements from the grade framework, which we call evidence grader, and actually is just about to be published. It's been accepted, so this is a preprint that's already up, where we're using the words in the article in the systematic review to help us estimate the grade scores. And we're finding that our method of actually taking advantage of the information that's available within the article is allowing us to do an okay, I wouldn't say fantastic, but a better job anyway of other methods, including random baseline, which is obviously not great, and a majority class classifier. So far, there's no competing methods to this, so we can't compare with anything else. But we're at least trying to wrap our heads around what it takes to do this kind of automated estimation of the quality. And although many systematic reviews do already use the grade framework in their assessment, many don't. And so the idea here again is about building a model that we can use to help us at least quantify the quality of evidence that's captured in a systematic review. And especially when we're looking at all the aspects of quality that are relevant to the grade framework and precision and consistency and directness and publication bias, some of these things are easier to detect than others. So risk of bias is a little bit easier to identify than inconsistency or publication bias, which is often difficult to see directly in the article. Okay, now on to discovery. So after we've structured the information that's in the literature and we've graded the quality of that evidence and synthesized it in some way. And by the way, I haven't told you about actually how we can generate summaries from literature. That's my student Yulia's work, which I'll save for another day. But once we've organized this information, we then want to make use of this information. So I don't want to stop at just organizing the information and making it easier to search for this information. I want to then build algorithms that allow us to make use of this information for discovery purposes. And so here I'm just trying to summarize some work that we did to generate hypotheses about drug-drug interactions and the opportunities for combining particular drugs for particular purposes. And this was actually done in the context of COVID-19 as well. And I think there's still a lot of room for improvement in this work, so I'm not going to focus too much on the actual sort of outcomes. But the idea here is to say, look, we can combine information from the scientific literature with information from other sources. In this case, we looked at clinical trials records. We can analyze these different resources, just like we can analyze the chemical patents with these concept recognition tools, bring them into the same kind of terminology space, and then we can start to do analysis over that structured information in a way that's almost impossible to do if you just work with the texts themselves. So we're really trying to organize the information that's in the papers and then use that for some kind of prediction or hypothesis generation. And there's lots of different algorithms that you can use to do that, including unsupervised approaches, mostly unsupervised because we don't know what we're trying to predict, right? And you can also take this paradigm and do something that's called literature-based discovery. And this is work that my student Grace C. Poo is doing and hasn't been published yet, so I can't tell you any details yet. Oops, sorry. But the basic paradigm is that we're trying to do prediction of interesting things that we might find in the literature by looking retrospectively. And so we're actually training machine learning models by collecting all of the concepts, the entities and the links between them, the relationships between them in a body of publications, and then trying to predict the future. And we simulate that by withholding the future, right? So we take data up until, let's say, 2020, and then try to predict what happens in 2021 or 2022. And because we can see what's been published and transform the literature in 2020 and 2022 into the same kind of entity and relationship diagram, we can see what new links form in the future at time t+1 that didn't exist prior to that point, and use that to drive predictive modeling, which allows us to make a guess, basically, of what are the interesting discoveries that are going to come in the future, which can be used to as a hypothesis generation tool, right? So if we have a method like this, which says, hey, there's this new connection, this new relationship between a mutation and a drug that looks promising, it may form the foundation of the next actual experiment to investigate that particular connection. So both the prior work and this work are really about trying to guess what are some interesting avenues for future research, but it's grounded in the literature and the evidence base that we have. Does that make sense? Okay. So, what do? The same process I've been telling you about all night. We take the literature, we identify the concepts and the relationships between those concepts. We build a graph that captures those entities and relations, and then we try to effectively do link prediction or edge prediction within that graph. And so once we're talking in this space here, we're not working with the text anymore at all. We're just abstracting the information that's in the text into this graph, and then doing analysis and modeling over the graph itself. Okay, I can't leave you today without talking about the other way that we can generate evidence. And this is something that's still more myth than reality, but it's something that I think is very promising and that we really need to be working on. And this can be framed in the context of what are known as learning healthcare systems, where we collect data from electronic health records, which represents the practice of medicine. We transform that into knowledge, and then we leverage that kind of structured information to drive this discovery process and produce more hypotheses that are tested, and then change perhaps the practice. And so we end up with this sort of virtuous cycle of driving the practice of medicine through to knowledge discovery to inform the practice of medicine, and we get better and better and better, right? We're collecting evidence, disseminating that evidence, and then changing practice, and then looking to see in the data what actually has happened once we implemented that evidence. And we can kind of overlay over this model a number of algorithmic and AI and machine learning sort of approaches which facilitate this whole process. Some very basic informatics, having common data models, having data sharing platforms which allow us to look at practice data at scale, and link together data from multiple resources, combining with algorithms like machine learning, and of course, taking advantage of distributed computing or cloud computing, having the outcomes of that being put into systematic reviews and publications, and transforming that information into structured knowledge, like the graph we were just talking about. And so there's a lot of research projects that you can overlay, which involve basically trying to capture data, transform the data, analyze the data, model the data, and ultimately return that data back into the evidence base. So this is a pretty sophisticated idea, but it's grounded in capturing of electronic health record data. And as soon as we start talking about electronic health record data, we have to start talking about text again. So you see, I always say that I have job security because there is ample text out there for us to keep working on. And indeed, by some estimates, about 80% of what's in an electronic health record system is actually unstructured data in the form of reports and notes mostly. So in order to do this whole paradigm, of course, we need to have data sharing infrastructure. We need to have interoperability so that we can compare data from different sources and different types of data together. And we need high quality data. And these are all areas of active research, not just by me, not only by me, not even by me in some cases, where the community can help us address how we're going to do this model and achieve this model. So imagine that we are able to connect and access and structure a huge amount of information. If you think about our PICO concepts and our chemical treats disease kinds of concepts, we can pull all of those things out of the electronic health record system as well. This patient was treated with this drug at this time, and this outcome happened. And at the moment, we're mostly doing this sort of thing by looking at structure data, but we can enrich that by looking at the notes and the reports and those sorts of things as well. And so the vision here is about organizing all of that patient data along a sort of timeline so that we can then start asking questions about what happens when you treat a patient with this disease and this prior history. What outcomes have resulted from that? So, and how do we collect the evidence? We can just count. That's like the starting point, right? Basic statistics. How often does something happen after a particular treatment? And then we can start to model it in ever more sophisticated ways. And, we heard about patient level genomics, and that's ultimately where we would like to go, right? Is to say a particular patient with this particular kind of data pattern can be predicted to have this outcome given this treatment. And ultimately, we hope to guide treatment decisions. So we can ask these kinds of questions of the data, right? Which treatment did patients choose after diagnosis? Which patients chose which treatments? That's about characterizing the patients in terms of their phenotypes, in terms of their data in the electronic health record system. How many patients experienced an outcome after a treatment? And then we can model, does the treatment cause that outcome, right? And does one treatment cause an outcome more than the alternative, by asking questions of the data? And the starting point for answering any of these questions is organizing the data, collecting the data, linking the data, and organizing it. And for all of that, we need modeling. And we can start by counting, but eventually we're going to need more sophisticated paradigms, which allow us to basically ask these questions of individual patients and answer them. So we really want to be using our models to support clinical decision making. Ultimately, a patient might have questions like, "Will I have another seizure? Will surgery help me? Am I depressed?" These are all things that our tools can help us to understand by comparing a patient to other patients whose history and outcomes we have access to. And just to give you a sense of a very simple model along these lines, we could try to predict 30-day readmission. This is a very common prediction task that people have worked on, and we would take all of the historical data that we have and then try to identify the probability of a readmission based on all of those factors. And the machine learning aspect of this, or the AI aspect of this, if you want to use that word, is about learning how all those variables combine and in what weights they combine. We can talk about all sorts of machine learning methods. And of course, you can't talk these days without talking about deep neural networks. And arguably, those deep learning models are needed because of the complexity of this data ultimately. One of my students, Xingwei Liu, he's actually wrapping up his thesis, and he's done some work on something called diagnosis-related group prediction. So diagnosis-related groups are estimates of the complexity of a particular patient that derives from what particular combinations of diagnoses that they have. So, in fact, in the Australian medical system, hospitals are reimbursed on the basis of the complexity of the patients that they deal with. It's not based at the individual, like we saw a patient with diabetes. It's we saw a patient with diabetes and I don't know, not a clinician. Anyway, it's a whole collection of characteristics, and that determines the complexity of the problem that they're having. Renal failure, maybe, or and so each one of these codes groups together a number of different diseases and phenotypes, and ultimately determines how much the hospital gets paid. And we started looking at the use of very early patient admission data to try to predict the DRGs for the patients in the mix, and found using our deep learning methods that we could do a fairly decent job of, even within the first 24 hours of admission, estimating what the eventual complexity of the patient case was. Ultimately, I'd like to see us moving towards a model or a paradigm that I call patients like mine, where we can imagine using these data science methods for characterizing and clustering or organizing the patients according to sort of similar histories and similar phenotypes. And so that if we have a patient that comes in, let's call the patient Miss Orange, we can place Miss Orange in the context of her most similar patients. And then we can use those data science methods to ask the questions of, okay, what happened to the other purple people? Miss Orange is a purple person. So if purple people have had this success with this treatment, maybe there's a good chance it will work for Miss Orange as well. And so there's this notion of sort of kind of dynamically identifying patient subgroups within the data that can, coupled with all of this sort of high level evidence analysis, can help us make decisions about how to treat individual patients. And we can combine all of these elements together to support particular clinical decision making, and bring together, the evidence that's in the literature. So you've got a patient, something's going on with Miss Orange. We've captured the data about Miss Orange, and we've looked in the randomized control trial space. We've looked at the evidence in the scientific literature. Maybe there's a guideline available. If there is a guideline, let's use it. If there's not a guideline, which there very often isn't, let's see if we can identify that cohort of similar patients and do some on-the-fly sort of modeling of outcome prediction to support a decision. Okay, so all of this is to say we need AI, and I would say arguably NLP, natural language processing, to enable learning from the data that's out there, both in the scientific literature and in the electronic health record data that we have, to ultimately support medical evidence generation, exploration, retrieval, and synthesis. And these tools can help us to find the information, to infer new knowledge, and to use the knowledge that we have to support our ever improving healthcare and clinical decision making. And with that, I'll stop and allow you to ask some questions. Thank you. Yeah.
Yeah, so I was wondering, you mentioned that with the complexity of these data sets, neural networks and deep learning models are probably going to be the most sophisticated options for analyzing these data sets. Do you think with that we might run the risk of doing black box like diagnostics and prognosis?
Yes. So I do think this is a major risk. And in fact, I have a whole talk and we published a paper last year, which was effectively an opinion paper, as a perspective paper in the Journal of American Medical Informatics, where we talk about the objective of replacing clinical decision making with AI tools, and specifically NLP tools in that particular context. And I think this is a very scary objective because, the one thing we can say about this paradigm is that the data sets never have the whole picture in them, right? We know they're incomplete. They don't have very simple things like patient values captured in them. If we're talking about, let's say, an end of life decision, perhaps there's a value system that's not going to be captured in the data that might influence that decision. If we're talking about a discharge decision, like, do you want to send your patient home? You need to understand whether they have family support or somebody who's going to bring them dinner. Because otherwise their risk of readmission is much higher. And so there's all these variables that we don't capture in the data, and arguably needs to come out of a conversation with a human being. And moreover, even if we had all the data in the data set, there is a human aspect to this, and sort of reasoning about different things that might be in conflict with one another, which I think would be very difficult for an algorithm to do. And certainly, is it black box? Yeah, I mean, these deep learning models are pretty much impenetrable. Although I will say that there's some opportunities thanks to language being the way that it is. So, I mean, you've all heard about ChatGPT. I can give another whole talk about ChatGPT, which I won't. But in this paradigm of actually being able to generate language from non-linguistic data. So I've done some work on medical report generation, where we're actually trying to generate descriptions of images. And it's very powerful, actually, to be able to verbalize in words that are shared and have a common understanding what's in an image. And so the black box of a computer vision or image processing algorithm kind of gets opened up a little bit if you can translate the features that have been recognized by the image processing into words. And so there's a very complex kind of interplay between these complex models and the capabilities of the algorithms. And I'm not sure where we draw the line, but I think we need to be thinking about supporting humans rather than replacing humans with these decision-making tools. Maybe I'll go there and then I'll come back to you.
First of all, it was amazing. Thank you for this. It is kind of related with the previous one, but is there any way that we can map our evidence to the reference, publications that we see, most likely in the?
Yeah. So I mean, if we go right back to the very beginning, sorry, this is going to take forever. If we go right back to the very beginning, we always know where it came from in this kind of paradigm, right? So we've processed the text. Hold on, let me find it. Which one am I looking for? I'm looking for a table. This thing, maybe. Ah, yes. Okay. So I can tell you. In fact, I've captured it here at character 0 through character 16, I have found this mention of this chemical. And so I can tell you the article where I found it and exactly where in the article I found the information. And so as long as we're indexing and storing that information, no matter what we retrieve, if we're using, the COVID-C model or any other kind of model, we can always go back to the source. And it's very important. And that's actually one of my criticisms of ChatGPT, because it's generating language. It's not indexing and retrieving language, right? And so the problem is that actually we don't have a source for much of that information. And that's a problem. And certainly if we care about evidence and truth, whatever truth is, scientific conclusions, let's call it, then we need to know where it came from. I don't trust ChatGPT to make decisions personally. I don't think you should either. Yes.
Yeah. So do you foresee whatever NLP model that dumps out from electronic health records data as being important, influencing what goes into generating the next set of electronic health records? Or is that kind of wishful thinking about how clinicians would actually use the NLP?
Well, so clinicians rely on evidence, okay, and they will change their practice if there is compelling evidence of a new treatment or a new understanding of particular counter indications for something. And so as long as we can convey and communicate that information in a way that they trust and they will respond to, then I think there is absolutely an opportunity to influence change, influence practice, and that it's that loop that I showed, right, where you have this kind of influence of what they're actually doing on what we can then learn and the kinds of studies that we can do. And of course, there's new stuff coming in, right? So the randomized controlled trials are always going to be adding new information in. And if we couple it with quality assessment and all of this, at the end of the day, we want to convince people that there is new knowledge that should change the way they practice. And I don't think, clinicians want to do the best thing for their patients. So if we're contributing to the knowledge and the evidence base that they use for those decisions, then yeah, we can influence that.
I guess more specifically, do you think it would influence like EHR systems to become more structured?
Oh, yeah. Okay. So was I telling you about, I don't know if I was telling Will about this. I was talking to somebody, I think I talked about it yesterday. So there was this ad for an electronic health record system back in the 90s when people were first starting to dabble with electronic health record systems, that had this great quote that I love. The clinician will point and click his way through the exam, because of course all doctors are men, but anyway, the clinician will point and click their way through the exam. And it's hysterical, right? Because what doctor do you know that will sit there and take a drop down menu with 2,000 ICD codes and be like, yeah, okay, that's the one I want? No. So, my opinion, we're never going to get rid of text, like documentation in terms of notes and reports and these sorts of things. It's all about human communication, human documentation. It's the way we think. People think in language, we communicate in language. So we're always going to do it in language. Now, there are aspects of it that are structured. Lab results, oh my God. Whenever somebody hands me a scanned piece of paper with a printout from a pathology lab, I want to pull my hair out and just say, are you kidding me? Like, this needs to be structured. It was structured in the beginning. You measured something, you know what you measured, you have a value, you have a unit. Like, just give it to me in structured data. We are making progress on that. So increasingly, our electronic health record systems are actually structuring information. And there's a bunch of activity, which I sort of sprinkled around the learning healthcare system model, to have common data models and to have a common language for representing the data. There's something called FHIR, the Fast Healthcare Interoperability Resource, which is essentially a way of having a standard for structuring messages that go back and forth between these electronic systems. So we're making progress on that, but it's definitely not done. And we're never going to get rid of text. So, whenever somebody says to me, oh, well, the solution to all of this is just not to have language in there in the first place, I always say, yeah, that's not the way people use language. That's how we communicate. That's how we document. So why try to force them into a paradigm that's not actually natural? There's a reason why we call this natural language, right? It's the way we communicate. I wouldn't be able to give you this talk by just showing you my slides, right? You need the words to contextualize. And that's exactly, I have another talk that I give all about this, but there's so much nuance that can't be captured with structured data. So if you look, maybe I'll just bring it up. Let's see if I can find it on the fly. Maybe not. But anyway, if you just look at a progress note, for instance, there's lots of subtle cues in there about, like the patient was mildly unwell, or the patient seemed to respond, or the patient had an acute reaction versus a gradual decline. And all of these kind of hedge words are really important for making clinical decisions and for capturing actually what's going on with the patient. And now, yeah, you could tell me that we could start to standardize that into some sort of like, okay, we're going to have a representation of uncertainty, and we're going to have a representation of, you know, the degree of a particular thing. But there's a limit to how much we can basically reproduce language and the generative power of language, productive power of language in a structured vocabulary. So why try when we have a thing called language? So, yeah.
I think when you have domain-specific knowledge that you're trying to represent, then the possibility, in a way, becomes more constrained, right? So if you think about the purpose of electronic health records is so one practitioner can communicate with another practitioner. So if the nuance, if the significant knowledge is all in the nuances, then that's not really conveyed properly either between physicians, if it's all very subjective, right? So I think in a way, when it comes, yes, language I think is full of possibilities, but when it comes to trying to represent domain-specific knowledge, the goal is to communicate across individuals about these specific knowledge, then there ought to be a situation where you can have a computer readable representation and a human readable representation, both in their sort of correct representation, but that are somewhat more correspond to each other than sort of complete wild possibility of natural language.
Sure, it's not completely open-ended. Absolutely. Hold on, let's see if I can find my little example. Yeah. So you're right that it's because it's a constrained context and a constrained domain. You know, to some extent we can anticipate and we can identify the patterns that are relevant, but there's a limit, you know, to what we can anticipate, because the variability and the variation is, there's so many degrees of freedom, right? And language is uniquely suited to expressing all of that variation, whereas our structured representations, and you know, what we're doing is very simple, right? We're trying to boil things down, entities and relationships between them. Actually, for the chemical patent stuff, we're realizing we're hitting a wall with that representation, and now we need to actually look at sequences of things, and we need to understand the relationships between them, between those events, as opposed to between the individual concepts. And so, you know, we can get increasingly complex, but at the end of the day, language is the most efficient way to communicate all of that complexity. And yeah, we can try to structure it as much as possible, but it's, you know, it's quite tricky. Let's see, but sorry, yeah. So, you know, this is a discharge summary, and there's obvious concepts that we can recognize, you know, the demographic information, the chief complaint here, shortness of breath and chest pain and chills and so on. But if you kind of read in, we cannot scan her chest, okay? We don't know why, it's not captured here, but for some reason they cannot scan her chest. And so they ordered a particular scan, the nuclear VQ scan, and then they decided that she was probably too unstable to go for that. So like we can see after the fact that you know, this particular test was done. We probably can't see that the particular test was not done, and we because, you know, we only capture things that were actually done, and the reasoning that's going on behind the decision making, like why was this test ordered versus that test, is not something that you can capture systematically in a structured data representation, which is positively oriented, right? And yeah, and there's more stuff here. You know, we've weighed the risks and benefits of anticoagulation, and given the possibility of a PE, a pulmonary embolism, it was felt that anticoagulation was very important. So like the reasoning and the, you know, that what this is capturing is a discussion amongst two people. I don't think we can anticipate all of that.
Yeah, so talk about AI sort of identify the superficial patterns without necessarily understanding the reason, and therefore when they make mistakes and make, grotesque mistakes because they fail to understand the reason behind.
Yes. Yeah. Exactly. It's not, it's capturing patterns. So we can talk about ChatGPT again. It's doing predictive text, but it's not understanding. It's not building an abstract representation of the factors that drive particular interpretations or inferences. Arguably, we need that if we're going to actually capture what's going on with people. All right. Way over time. Oh gosh, the pizza talk. Yes. Okay. Thank you all. And for those of you in the room, let's have pizza. And for those of you not in the room, I'm sorry you don't get any pizza. Thanks for listening. Okay.